last speaker of the session. She's Leighton Durham. She's a second-year PhD student in clinical psychology at Vanderbilt University. And today she is going to talk about how global brain volumes relate to general psychopathology in children. Right? Welcome, Leighton, on stage.
Let me go ahead and try to share the screen, make sure it works okay. Okay, are you seeing the
Go ahead. Thank you.
slide? Okay, great. Well, hi all that are here, and thank you for your presentation. Those are awesome. And thanks for the introduction, Alex. I'm excited to have the opportunity to present today. As Alex mentioned, I'm a second-year graduate student in a PhD program at Vanderbilt University in Nashville, Tennessee in the US. And I'm working under the supervision of Dr. Antonia Kazkirkin. In her lab, we investigate neural mechanisms of dimensional psychopathology. And we're particularly interested in internalizing disorders and internalizing symptoms. So today I'll be talking about the relationship between global brain volume and psychopathology in a child sample. And those listed on the title slide here below are those that helped with these analyses that I'll be talking about. Some are from Vanderbilt, others are from University of Pennsylvania and University of Chicago. Did that switch slides okay? It's all working okay? Okay, good. All right, so just to start with a bit of background, childhood is a critical time for both brain development and for the manifestation and onset of psychopathology. And importantly, psychopathology during childhood is a considerable risk factor for later psychopathology. So it's critical to promote earlier detection and prevention of psychopathology at an early age. Also, many forms of psychopathology have high comorbidity rates, and there's an increasing body of literature showing that symptoms are often dimensional, shared across different diagnoses, and hierarchically organized. There's also a substantial degree of heterogeneity within specific forms of psychopathology, so disorders don't look the same for all individuals, and they can present very differently. However, most research surrounding psychopathology uses a more categorical approach with a case control design. So for instance, a DSM-defined diagnostic group compared to a healthy control group. And the primary issue there is that you lose a substantial chunk of the spectrum of psychopathology as it manifests in reality. So anyone who doesn't quite fit into either the case group that's specifically defined or a more clean, healthy control group, however that's defined in a given study, and whether that's due to subthreshold symptoms or comorbidities or an alternate clinical presentation, for instance, those people get lost and are not considered in those type of designs. Some prior studies that have taken a more dimensional approach have shown negative associations between psychopathology and brain volume in youth samples, but the majority of them have used samples with relatively wide ranges for age, and that can obscure developmental trajectories given the amount of change in both psychopathology and brain development that's going on during childhood and adolescence. So in the current study, we aim to examine associations between regional gray matter volumes in the brain and forms of psychopathology in children from a dimensional perspective. Our analyses build on prior work primarily through a larger sample size, a more homogeneous age group, and a dimensional approach to measuring psychopathology, which could reflect the true nature of psychopathology more effectively. Our primary hypothesis based on prior studies was that general psychopathology would be associated with smaller regional gray matter volume, and that would be the case throughout the brain in a relatively non-specific pattern. So the sample for this study came from the ABCD study, which stands for the Adolescent Brain Cognitive Development Study. If you're not familiar with it already, it's a large longitudinal cohort study of brain development and child health. And the data is collected all over the US and is publicly available. So the participants for this specific set of analyses all came from the first wave of ABCD data collection, which includes children at 9 to 10 years old. And they're recruited at 21 sites across the US. The total sample size for this baseline wave is 11,875. We ended up using the final sample for these analyses of 9,607 after rounds of exclusions for lack of imaging data or missing data on other variables we needed, like psychopathology or covariates, or failure to pass relatively stringent quality assurance measures for the imaging data that occurred on the ABCD side. And the sample was split pretty evenly across sex, and the racial ethnic breakdown is listed there as well, as about 53% white, 20% Hispanic, 14% African American, 12% other. In terms of the measures we use for these analyses, we use data from a psychopathology measure called the Child Behavior Checklist for school-age children, which is commonly known as a CBCL. And it was a parent report measure about child behavior. So the individual items ask about behaviors that could be of concern or psychopathology symptoms and are rated on a 3-point scale, from 0 being not true or never true, and 3 being often true or very true. Brain volume was measured using 3T MRI imaging, and that was conducted at the 21 data collection sites. And regional GMV was defined by automated atlases, one for cortical regions and one for subcortical regions. The image processing was performed on the ABCD side by the ABCD Data Analysis and Informatics Center, and they used centralized protocols and approaches for all sites so that it was consistent. And for statistical analyses, we started with factor analyses. An exploratory factor analysis of 66 CBCL items in a random half of the sample was performed by some of our collaborators at University of Chicago and University of Pennsylvania, Tyler Moore and Ben Lahi. And they identified 3 dimensions of psychopathology that they labeled internalizing, conduct problems, and ADHD symptoms based on the items that we're grouping together. And then a confirmatory bifactor analysis with the second half of the sample was used to model those 3 factors plus a general psychopathology factor. And that represented the shared variance across the 3 factors. For the brain volume analyses, structural equation models are performed to look at associations between volume and those 4 psychopathology dimensions. And we focused on regional volume of 68 cortical regions and 19 subcortical regions. A couple of other important things to note here are that post-stratification weights were provided by ABCD and applied to account for the stratification of the sample across different data collection sites. And data was also clustered by family, since the ABCD study includes some twins and other siblings. The model we tested for each brain region was listed, was as listed there on the slide. So the covariates of note interest that we included were age, sex, race, ethnicity, and MRI manufacturer, because there were a few different scanner models used. And then we, of course, included the psychopathology factors as well. And to control for multiple testing across all of these different brain regions that we were looking at, we controlled for the false discovery rate at 0.05. All right, as I mentioned briefly, the confirmatory bifactor analysis of the CBCL data in this case yielded 4 orthogonal factors of psychopathology. So the factors are totally uncorrelated, which allows us to look at more distinct relationships and phenotypes. General psychopathology represents the shared variance across all items. So you can think of it as representing the symptoms common across multiple disorders and domains. And then 3 specific factors for internalizing, conduct problems, and ADHD symptoms represent what's unique to that domain once that shared variance is extracted. So each CBCL item loads onto exactly 1 specific factor as well as the general factor. An example of some of the items that load onto internalizing, conduct, and ADHD are listed there for those that are interested. So now I'll just summarize some of the results, and it's organized by psychopathology factor, just for clarity. So the general psychopathology factor, as we expected, was found to be, we found that increased general psychopathology was associated with smaller regional GMV throughout the brain. And this was a pretty global effect. Out of the 68 cortical regions we tested, 54 were significant. And that's after the FDR correction. And of the 19 subcortical regions, all 19 remained significant after correction. And this figure is just a visual representation of those findings for the cortical regions. The brain map shows the majority of cortical regions were significant, and the scatter plot shows that as scores for the general psychopathology factor go up, cortical brain volume goes down. For the conduct problems factor, we also found a relatively global effect that was similar to that of the general factor. There were significant inverse associations for 52 out of the 68 cortical regions and 15 out of the 19 subcortical regions. And again, that was after correction for the multiple comparisons. And this figure again just shows the map of significant cortical regions and the scatter plot of cortical volume against extracted scores for the conduct problems factor. For the ADHD factor, there was still an inverse association, but it was not as global in nature as that of the general and conduct factors. 25 of the cortical regions were significant and 8 of the subcortical regions were significant. Interestingly, we didn't find any significant regional results at all for the internalizing factor. So we didn't see the same pattern there with that factor, which I'll mention again and explain some reasons why we might think that's the case. So just a few discussion points to go over. These results are consistent with prior work that shows general psychopathology associated with smaller gray matter volumes. And it also extends prior work by showing that these associations are apparent at this young age of 9 to 10 years old, and that they show up for specific conduct problems and ADHD factors as well. Some additional relevant points from the literature to mention are that prior work has shown a general factor of psychopathology to be associated with an array of functional deficits, so including areas like self-control, emotional regulation, and executive functions. And studies have also shown reduced functional activity at the whole brain level across multiple categories of disorders when performing tasks involving cognitive, social, and reward and threat processing. So these findings all are compatible with our observation of nonspecific smaller GMV across regions involved in a wide variety of functions. And as I mentioned briefly, our lack of findings for the internalizing factor is interesting, and we suspect that this could look different at a later age. Internalizing disorders become more prominent in adolescence, so it's possible that associations between internalizing symptoms and brain volume aren't showing up quite yet and will at a later time point. It's also possible that the any associations that could be there between internalizing symptoms and brain volume are occurring through the loadings on the general factor, to the point that there's nothing left for the residual specific internalizing factor to explain. So this will definitely be an important future direction for us when we have more waves of this data to look at. Some primary limitations I just wanted to mention of this study are the cross-sectional design primarily and the parent report psychopathology measure. Fortunately, we're going to have, with regard to the cross-sectional design, we will have plenty of follow-up data later on because this sample they'll continue to collect data with this sample. So down the road, we'll be able to sort of get a better understanding of these developmental trajectories in the areas of the brain, as well as in psychopathology in their true form, and look at temporal sequences more closely. And for the parent report psychopathology measure, this is interesting because some people would argue that parent report is better and some argue that self-report's better. These children are 9 to 10, so they might be at the age where they could report accurately about their own psychopathology, but all we have access to at the moment is a parent report measure. And this slide just lists some summary points and conclusions. So I'll run through them briefly. I'm not sure how I'm doing on time, but consistent with the emerging notion that the general factor may relate to non-specific variation in the brain, these present analyses provide support that general psychopathology symptoms observed in childhood are inversely associated with global gray matter volume. Reduced gray matter volume might be a transdiagnostic risk factor across psychopathology domains and disorders, but future waves of data will allow us to delineate these relationships further and help us establish a better understanding of the timing. Smaller brain volume may also represent a risk factor for ADHD and conduct problems more specifically, but we found no evidence for internalizing symptoms in this current study. And finally, considered together, these results support and expand upon the prior work on the relationship between neurostructure and psychopathology from a dimensional perspective in childhood. This just lists some acknowledgements, the contributors again for these analyses, some funding information, and the ABCD study statement. And thank you guys so much for listening. I appreciate it. And thank you for the opportunity to present today.
Thank you very much, Leighton, for your presentation. Yeah, we have just a couple of minutes for questions. I think at 50 past, we're going to be kicked out automatically because there's a keynote session starting. But we have a few questions from the audience. So the first one is by Sudhakar Tumala. They are saying, nice presentation. Which region showed highest association? And what is the correlation with the amygdala?
That's a great question. So that's actually going to be my next follow-up project, is to look more closely at the regional results, because it was so global that we didn't investigate the region specifically quite at the level of detail that I would like to. So there will be more to come on that, but it's a great question. We're in the process of figuring out ways that we want to look at more like clusters of ROIs and which regions are most correlated with which dimensions, and looking at it from that direction. So that's a great question.
If there's no time, I will send you the questions later.
Okay.
So Vlad is wondering if it would be interesting to look at cortical thickness or surface area to get a feel for which accounts for the reduced gray matter volume.
Yeah, absolutely. That's also a great question. We actually had a student in our lab looked at basically the exact same thing with cortical thickness measures. and found, which this was very surprising to us because they're typically pretty highly correlated, volume and cortical thickness, and she did not find near the amount of significant results that the volume analyses did. So we're not really sure why that's the case, and it would definitely benefit from further investigation, but that's a great question. We have looked at several modalities, and volume definitely seems to be the strongest by far, which is interesting.
I will take a last question before we are kicked out. So Sin Kim is saying, if I recall correctly, doesn't GMV tend to decrease in general during adolescence? Perhaps another evidence to the hypothesis that early maturation can be harmful.
Yeah, and that's also a great question. That can be really relevant to childhood trauma as well. We have a student in our lab looking right now at the impacts of trauma on brain volume and cortical thickness at this age. So that will be interesting to see what that looks like in terms of the trajectory of gray matter volume development and then decrease in adolescence. So I think that's a great question. I think the future waves of ABCD data will be extremely helpful in looking at different trajectories like this, because it's certainly, there's certainly a wide variety of individual differences. And it's nice that we have this specific age group to sort of be able to pinpoint, you know, specific relationships at a given age where the variety might not be quite so broad. But it also means that we can't say much beyond what's happening at 9 to 10 years old. So I think having the adolescent waves will be extremely impactful so that we can, you know, do follow-up analyses and look at what this means for adolescents in this sample.
Okay, thanks, Leighton. I need to wrap up the session. I want to thank again Anis, Riane, and